and as will be demonstrated by this project for innovative biotechnological exploitation. SmartX project is coordinated by Marco Giovine with the University of Genoa for the Italian group, and from Misha Aylan from the University of Tel Aviv for the Israeli group. The project will be presented, if I understand well, by a single presentation shared by Misha that will be the first one to present, followed by Marina Pozzolini, Sonia Scarfi, and will be closed by Marco Giovine. You will have in total 1 hour, including the questions.
There is a demand for it.
Sorry? Marco, you are saying something? No. Okay.
No? It seemed Valerio.
So,
No, my microphone is off.
Okay. So I will switch on my camera when there will be about 10 minutes remaining at the end of your time, just to keep your time enough. And please, Misha, you can share the screen and start.
I do hope it will work. Open. Oh, here it is. But I don't see it. Do you see the presentation?
Yeah.
I do.
Yes.
Okay. So just a second. Now you have it on all full screen? Yes.
Yeah.
Okay, thank you. A bit more than a year ago, we've been gathered in the Festival Mare in beautiful Genova to present starting points or mid-points of our project, a SmartX project, which is sponge mariculture for innovative biotechnological exploitation. As you've heard, the Italian group is headed by Professor Marco Giovine and myself in Tel Aviv in Israel. And what can I say? Now, this was the opening slide, and now, a year and a half later, our meeting is through, not Zoom, but electronic, and the world we knew turned upside down, and maybe now we are starting to see some bright future with the vaccine on the way. So I will try to show you what we've done. My group has several interests, and much of these interests are engulfed and are within the project we've seen, whether it's regenerative medicine, biotechnology, and obviously drug discovery, and the center are sponges. And we study them on all aspects. But within this framework, I just wanted briefly, very briefly, because I thought it would be to the general audience as well, to tell you what are sponges, but I will not repeat what most of you heard last year. Sponges are sessile organisms. They are sessile animals. Actually, the first animals to arrive on this earth, on planet Earth, are sponges, and they are filter feeders, which means they pump in water and feed on whatever is inside, small particles the size of bacteria and even viruses. And that's what they feed on. They are a huge group, beautiful, everywhere where there is water. And the largest class of sponges are called demosponges or demospongiae. And they have a skeleton which is mostly made of silica spicules, the demosponge, siliceous spicules, and if they have organic skeleton, it's made mostly of collagen. Some of the species also chitin. So this brings me to what we would like to show and to study. And collagen, as you probably know, is a family of protein, the most abundant in any animal, including human body. And our aim was to try and evaluate if we can find commercial applications out of this natural product, either for cosmetics or for therapeutic regenerative medicine, and also maybe for drug discovery. I bring these two very recent papers, collagen-based materials in cosmetic application. Both are from 2020, you see, September 2020. And there are many potential application for collagen. And there are problems with the common collagen we find. Another aspect is that sponges are very well known, at least among scientists, to be an extremely rich source of natural products. Actually, the most prolific source out of marine environment. And these natural products could be for pharmaceuticals, but also cosmeceuticals and nutraceuticals, etc. And I really like to show this review, the Marine Biodiscovery Pipeline and Ocean Medicine of Tomorrow, because we are hosted, although electronically, in Naples, and Adriana Ianora is the leading author on this paper. But as you can see, many of these compounds come from sponges like this, which is actually the first medicine to come out of marine sources. This is both antiviral and anti-leukemia. But there are others, and each year, certainly in the last couple of years, there are more and more compounds. We would like to take advantage of the fact that sponges have a natural capability of multiplying and reproducing themselves asexually by fission or by branches or other modes of asexual reproduction, and they regenerate, so it's very nice to use it. And these are prototypes, by the way, from the Red Sea, not from the Mediterranean, which we try to culture different, in this case, in the last one, 4 different species in situ in the marine environment. But what we are now trying to do is to go to another sponge, this time a Mediterranean one, which it is the star of our research, Chondrocene reniformis. But unlike what I told you earlier, it doesn't have siliceous spicules, which is good if we want the collagen, and no large fibers, but many fibrils of collagen. And the research was aimed to optimizing mariculture or aquaculture of this sponge in order to get its collagen and to evaluate it. And one of the leading reasons was this map of the Mediterranean, which is a heat map to show the surface temperature. And you can see that in the west part of the Mediterranean, Italy, it's much, much cooler than in Israel, in the eastern Mediterranean, in the Levant. And this is actually good for our purposes because the hypothesis was that if it thrives, the sponge, here in the East Mediterranean in high temperature, its collagen would have higher thermal stability than collagen built in cooler or much cooler water, and higher thermal stability is very in demand if we are talking about some product to be used in human body. So that's where we are in Israel. And the specific goals of this joint project, as we discussed in 2019, were the applicative aspects of the sponge and how can we exploit it. And we gave several tasks. Okay. Yes, the tasks were to look for sites, depth, to characterize the collagen from different places, and to try to evaluate its role in different aspects that we would like to check, either as protein or as compound, and to get biomass of high quality collagen. That was actually our purpose. So what have we done? First of all, we had to collect the sponges. Well, we wanted to do it from two source of population. One, which is the shallow water, where it's extremely hot in Israel. It can reach 30, 31, and in recent decade also for an extended period of time. So whoever lives there must cope with the temperature. And the other one is from mesophotic, deep water, around 100 and more meters, but this is being done with an ROV, with a robot, a remotely operated vehicle from a research vessel, because it goes out 15 kilometers offshore. And there, the temperature is almost stable, around 17 Celsius, what you can find also in Italy. This higher picture I simply stole right now during the talks of our earlier talks, yes, because it also shows the heat map of the Mediterranean. And if we look, this is the East Mediterranean, the Levant Basin, and obviously you all read Hebrew, so you know that this is the Mediterranean, and these are our locations where we worked. Collection sites and culturing sites. East Mediterranean, this is Tel Aviv. Okay. And what we've done, we went out, we collected. It's not an easy task with the robot. And we always also left some parts on the rock in order for it to regenerate the natural population. But we took within a bag sealed to the lab, and then we had to acclimatize it to our system. So we had tiles, which this is my master's degree student, Boaz, who drilled in order to have on each tile a specific specimen, all marked, and he just put it on the tile, and the sponge very fast, it could be sometimes within 24 hours, adhere and attaches itself to the tile. So, and this is in a running water, flow through water system in the lab. These are two, maybe you know, Agellas oroides, Sacotagus, Spinosulus, other sponges. Okay, and then the idea was, okay, let's try and see whether they live either underwater or inland. So underwater, what have we done? We took these tiles, you see, the sponge adhered to it and lived at least a week in the running seawater, and then we took it to the field. In the field, before we brought the sponges already drilled into the rock at 10 meters were these boxes, which we could also close from above. And the idea was that we can, by this way, also reduce the amount of light, which other works already have shown that they may interfere with the growth of the sponges, and also lower a bit the currents, because in and siltation, because in shallow water, they usually are, in Israel at least, they live in crevices. Only at 100 meters, where it's the most abundant sponge, they live on the surface. But their light is much less. So we had to reduce light and some of the currents, and that's how we did it. Also, it protects from potential large predators. So this is the setup. And in each box, we had 6 sponges, 3 taken from the shallow water, 3 from the deep water, and all brought to 10 meters depth to see what's going on with them, how will they live, and we had these 5 repetitions. Okay. That's how it looked from above. Here, this is the scheme. Also, we put here a hobo in order to get all the physical measurements like light, etc. What was incredible that we didn't lose a single specimen for the entire 3 months of the experiment. All of them survived. But what was nice to see that the sponges that originated from shallow water in the blue color, they grew faster and larger than those from the originated from the mesophotic, from the deep, cooler water. But also, you can see the seasonality, because the outside, the ambient water within something like less than 50 days rose from 22 Celsius to 29. So the ambient water warmed, or actually heated, and then it stays even. The temperature went even higher and only then reduced. So also the growth showed a negative inclination. This is because the sponge, if it grows like this. But when it shrinks, it usually does something like this, and then maybe even separates to smaller sponges. So this is why we see a decline in size. But please remember the growth, and especially of those which originally came from warm water, the large growth. But none of them died. Here it is around 29 degrees, 60 days, 2 months. And growth of those are nearly 90% within 3 months. So we saw a trend of seasonality, and we saw a significant difference between the two groups or sources of chondrosia, either from cooler water or from the shallow warm water. But successful transplantation has also other implications. For example, I will not go into it, but will the environmental condition change for the better, then we may also use deep water or mesophotic sponges in order to replenish the population in the shallow water. The aquaculture, the indoor experiments here, these are all aquaria with sponges and in 3 sets of different temperatures. Again, Boaz here taking picture. What does he do? He placed these sponges now on black perspex in order to get better picture. This is one of the things he learned from the in situ experiment in order to get a better picture, because he used a software to measure their size. So when they are on tiles, it's a bit more difficult to see the exact contour. So on black, there is a much better contrast, and the pictures can be assessed better. Because earlier we saw that around 25, 26, it's the best increase in size. Therefore, he chose 3 temperatures, 25 as, let's say, optimal, 31 as the highest temperature in the sea that will be, and ambient. However, the ambient, you have to remember, it changes during the course of the experiment. So it rose from around 17, which is winter temperature, to 26. Again, all individuals survived, which is beautiful. For maybe you are not aware of it, but to culture sponges indoors, even in flow-through system, it's a huge task. But this animal is vicious. It survives so well. And at the beginning, we saw the increase in size, but also we increased the water temperature very, very slowly. It took us half a degree a day until we reached the final goal, whether it's 31 or 25. That's why we are lacking here. And then the curves show the size, relative size, to the original size measured when they were placed in the experiment. And you see, those which were cultured in 25 degrees increased in size. There is some shrinkage here, although you see the standard error, but increase in size to something close to 60% from the initial size within 75 days or even more, whereas those which were in hot water, they got into about 35% of the initial size, but then when the temperature was consistently 31, they started to shrink, but didn't die. And the one that were in ambient temperature, well, that's an explanation. The ambient temperature rose from 17 to 26, actually resembling the one of the optimal growth. So they cut with their size with the group of 25. So we see a significant, when I say significant, obviously it's all statistically tested and everything. significant difference in growth rate between 31 degrees and the group that was cultured in 25 degrees, the treatment. And or maybe I should have said it earlier, they can be cultured indoors, on land with running seawater and survive for at least 3 months. So this is a huge take-home message from this culturing experiment, both in sea and in indoors. And then Boaz flew to Genova to conduct further characterization of the collagen, which my Italian colleagues will tell you. I show it here, but I'm sure that Sonia will talk about it, so I will leave it aside. But I have one more slide to show you, which is because of these experiments, we got tempted to do further work, and this is a huge setup to test the different diets impact. You see here the bottles with different diets, which was provided to all these aquaria with a sponge, in this case, Sarcotragus spinosulus, also a sponge without siliceous spicules, but with collagenic fibers. And this setup will be used also, a similar setup, to test the chondrosia if we can get a better diet for their growth. So to get an optimal temperature, an optimal diet, combine it, and hopefully get a way to do it indoors, on land, throughout the year. Because remember, out in nature, they have their own cycle where they reduce in size once the temperature arrives around 29 degrees, 28. First they stop growing, and then they reduce in size. And then when it's cooler again, they start to grow. But we would like to have only the part where the curve is constantly growing rapidly. And if we can get it into a culturing system, this could be an agricultural product to be used further down the line. So here I will thank my Italian colleagues with whom we had all the time corresponding. We also visited both ways, but then came this nasty virus and stopped us in the track of the mutual visits, but didn't stop our research. Even during shutdown, we attended the animals, otherwise they would have died. So we got special permission to go and check and monitor and feed and everything. And in Israel, there are many students, colleagues that helped. And this is the place where we had the running still have the running seawater system, the ship and ROV, and of course, the funding agencies. And I will end the show now, and I don't know if you want now questions or you want to first continue.
Maria?
Yes, there is now Marina Pozzolini that have to continue the presentation.
So we will have the questions at the end?
Yes. So as for the other...
Just tell me if I took out the share screen. Yes?
Hi, Marina.
Yes. Okay. Good morning, everybody. I'm very excited to be here at this webinar. And now I try to keep the slide. Okay. Can you see? Okay. I will tell you some our results regarding the task 1 and 2 of the SMART project. And that are regarding the sponge collagen isolation and its chemical characterization. So, as Micha told me before, we are interested to isolate collagen suitable for tissue engineering. And the task 1 and 2 of our project was, in particular, the selection of the site, the best site for sponge collection, and the second task was the extraction and biochemical characterization of this collagen. We know that the collagen more suitable for tissue engineering should be provided with a good thermal stability, and it's known that the thermal stability of the collagen is related to the level of the hydroxyproline residues in collagen. It's also known that the marine collagen is characterized by lower thermal stability than the mammalian collagen. And this is due to the lower level of the hydroxyproline residues. So, as Micha told me before, our hypothesis was that maybe the thermal resilience of the Chondrocene reniformis sponge that is able to live at a different temperature could be due on its ability to adapt the collagen fiber or maybe other extracellular component in the body to the seasonal temperature variation. So we asked a question: could the collagen isolated from sponge recovered in summer at high temperature be more suitable for tissue engineering application than which isolated from sponge recovered in winter at low temperature? And in order to answer at this question, we need to compare the thermal stability of the collagen fiber isolated from sponge specimen sample in summer and in winter. For this purpose, in this project, we received two groups of Chondrocene reniformis specimens from the group from Israel. One group was collected sample in winter, in December, in 2017, at 70 degrees, 3 meters of depth. And a second group of specimens was collected in the same place, in Est coast in Israel, in summer, in July, in 2018, at 70 degrees, at the same depth. Then we received this specimen frozen, and in our lab in Genoa, we purify the collagen fiber using a validated protocol that we have previously described in our paper. Using this protocol, it is possible to obtain a large amount of collagen fiber, very purified. It's like a fibrillar suspension, viscous and dark. And as you can see in this picture of the electron microscopy, this suspension is mainly formed by collagen fiber. The 95% are protein, mainly collagen, and there is present a little percentage of glycosaminoglycan inside. And then, using this protocol, we purified collagen fiber from the sample collected in winter and the sample of sponges collected in summer. Then we compared the thermal stability of these two kind of collagen fiber. Using the DSC approach, the differential scanning calorimetry, this is the more suitable approach for the analysis of the thermal stability of the collagen because it is suitable for the collagen organizing fiber. And using this approach, we analyze the stability. Here you can see the results. This is a replicator of one analysis of the thermal stability of the collagen fiber isolated from the sponge collected at 70 degrees in winter, and is, as you can see, the thermal, the denaturation temperature of the collagen fiber was an average of 72 degrees, while the denaturation temperature of the collagen fiber isolated from sponges collected in summer was an average 10 degrees higher. So this analysis confirmed that the collagen derived from sponges collected in summer has provided with higher thermostability. In order to explain this behavior, the molecular reason of this change in thermostability during the changing of the season in the sponge, we analyzed some different biochemical parameters. The first was the analysis of the amino acid percentage. As you can see here, we have the percentage of the different amino acid in the collagen fiber isolate from sponge collected at 70 degrees and 27 degrees. This is the typical profile of amino acidic profile of the collagen with high level of glycine and the presence of hydroxyproline and proline. Here we have the, in this picture is the representation, the graphic representation of this data. And as you can see, we didn't observe any significant variation in the amino acidic composition in the two kinds of collagen. However, when we calculate the hydroxylation ratio, so the number of the hydroxylated lysine and hydroxylated proline, we observe a little but significant difference in the level of the hydroxylated lysine amino acid in the sample collected at either temperature. The hydroxylysine in collagen is involved in the fiber aggregation and maturation. It is involved in the cross-linking of the fiber. We then analyze also the content of glycosaminoglycan inside the fiber, but as you can see, we didn't observe any significant difference in this component, and the amount of glycosaminoglycans in this fiber resulted lower than which observed in the fiber isolated from specimens collected in Italy. As we observe an improvement of the level of the hydroxylysine in the collagen fiber collected, derived from sponges collected in summer, we detect also the level of the global glycosylation level of the collagen fiber, because very often in the hydroxylysine residue are present glycosylation in the collagen, and also is known for many years that the collagen derived from Chondrocene uniformis is characterized by high level of glycosylation. So we wanted to detect the level of the glycosylation in the two samples of the collagen fiber in order to understand if this parameter could be involved in the thermal stabilization of the fiber. And we observe a significant improvement of the glycosylation level in the collagen fiber isolated from sponges collected in summer at 27 degrees. So here we have a short summary of these results. The collagen fiber isolated from sponge specimens collected in summer at either temperature has higher thermostability, and maybe this thermostability improved is due to the improved level of the hydroxylysine and the improved level of the glycosylation of the fiber. We didn't observe any significant difference in the glycosaminoglycan in the fiber. Then we also tested the mechanical properties of the membrane produced using this kind of collagen extract derived from sponges collected at 70 degrees or at 27 degrees. The mechanical properties was evaluated using this method, the MTA, Dynamic Mechanical Thermal Analysis, in which the membrane were subjected of mechanical stress, improving the temperature. Surprisingly, we observed that the membrane produced using the collagen fiber isolated from sample of sponges collected in winter resulted more resistant than which obtained prepared using the collagen fiber isolated from sponges collected in summer. And this mechanical property was maintained better in this sample. This improvement of the mechanical properties in this membrane could be related in an improvement of the diameter of the fiber or in changing in the texture of the membrane. So we analyzed different regions of this membrane by electron microscopy, but we didn't observe any significant difference in this parameter. At the end, we analyzed also the biocompatibility of this membrane in fibroblasts and in keratinocyte cells in terms of adhesion and in terms of proliferation. And as you can see, we didn't observe any difference in the membrane produced with the collagen fiber isolated in winter or isolated from sponge collected in summer. They maintain the already described biocompatibility of this kind of membrane. So in conclusion, we can see that the collagen fiber isolated from sponge sample in summer display higher thermostability than which isolated from sponge sample in winter. And the increase of thermostability could be due to the increased level of the lysine hydroxylation, not the proline hydroxylation, and the increased level of the collagen glycosylation. Conversely, we observed that the membrane produced using this kind of fiber presents either level of stiffness when we used the fiber isolated from sponges collected in winter. And to explain this peculiar behavior of this membrane, maybe the answer could be the peculiar behavior of the Chondrocene reniformis. Some specimens in summer has this peculiar behavior to creeping their body as asexual reproduction, and maybe the collagen fiber in summer could be less reduce the mechanical property in order to obtain this effect. So this is all. Okay.
Okay, now we pass to Sonia Scarfi.
Sonia.
Yes.
Okay. Good afternoon to everybody. I will try to share my screen. Okay, can you see my screen?
Yes.
Okay, yes. Thank you very much. Okay, so I'm from the University of Genova, and I developed the task 3 of the SmartX project, which is based on earlier observation made in our laboratory about the behavior of Chondrocene reniformis. We had observed a particular cytotoxic activity of this sponge when it was hosted together with other organisms in the same tank, in which the sponge was able to kill the neighboring organisms. So we hypothesized that there could be the release of some toxic compound in the water, killing the other organism. And our idea was to try to understand if this toxic compound could also have anti-tumor properties. And for this reason, we developed the task 3 of the SmartX project. So the task is about the extraction and step-by-step biochemical purification and final identification of Chondrocene reniformis anti-tumor proteins, and to assess the degree of anti-tumor activity of the product of the sponge during the purification, and by comparing the cytotoxic activity on normal cells against tumor cell lines, and also trying to understand the mechanism of action of this compound. Okay, but why am I talking about a cytotoxic protein? This is due to the preliminary observation that we made when we tested the cytotoxic activity of a simple crude extract of the Chondrocene reniformis sponge that was simply obtained by cutting the tissue of the sponge and squeezing it. This is the result of the cytotoxic activity that you can see here in this cell line, but it was repeated in other cell lines also, where we have a high rate of mortality. So we try to understand, first of all, which could be the chemical nature of the compound or of the group of compounds able to give this cytotoxic activity. And first of all, we performed a dialysis separating high molecular weight compounds from low molecular weight compounds of the crude extract. And as you can see here, we saw that almost all the cytotoxic activity was retained in the high molecular weight fraction of the crude extract that is above 10,000 kilodaltons, that is here. So this already pointed out some macromolecule or some big molecule having this activity. So the next experiment was a thermal inactivation of this dialyzed extract to try to understand if the compound was thermolabile. And in fact, after this thermal inactivation, we could see that almost all the cytotoxic activity was lost in the dialyzed extract. And finally, in the other experiment, we tested the hypothesis of the protein, of the cytotoxic protein, by digesting the dialyzed extract with different concentrations of trypsin that you can see here, a lower concentration and a higher concentration of trypsin. And again, also in this case, we could observe that the cytotoxic activity was almost completely lost after this digestion. So this, of course, pointed out to the activity of one or more protein with cytotoxic effect on the cells. So we performed a step-by-step modification that I don't have time here to explain, but the last step allowed us to perform a 2D gel electrophoresis of some specific protein fraction and an analysis by mass spectrometry that allowed us to finally identify the protein responsible for this activity. These are the last step of the purification and the identification of the protein. This is the chromatogram obtained by HPLC separation of the protein fraction by molecular weight size exclusion. We obtained 5 different peaks that were tested for the cytotoxic activity, and we found out that almost the activity was retained in peak 4 and peak 5. So these 2 peaks were subjected to 2D gel electrophoresis, and the spots were digested with trypsin, and afterwards mass spectrometry analysis was performed on these digested spots. The peptides that were retrieved and identified were matched with the proteomic database of Chondrocene reniformis that we already had in our lab. And we saw that almost all peptides from all these spots corresponded to this protein here that you see, retrieved from the Chondrocene reniformis protein database, that is a low molecular weight protein, completely unknown, of 19 kilodaltons of weight, and more or less 200 amino acid long. An analysis of the structural homology of this protein pointed out that there were the presence of 2 domains with a high homology with 2 already known proteins. One of these domains is highly similar to the ryanodine receptor calcium channel, while the other domain is highly similar to the defensin beta sheet domain. So this slide is quite complicated, but I'll try to summarize the results. These are all the cytotoxic experiments on using the various fractions during the different steps of purification. In the final histograms here, we have peak 4 and peak 5 that comes from the HPLC purification. And there is a comparison between the toxicity on normal human fibroblasts, so normal cells, and on tumor cell lines. In the center, we have 2 murine tumor cell lines, while at the right, we have a human tumor cell line. What we can observe here is that there is a certain degree of toxicity, but not very high, in the human dermal fibroblast when the protein is purified in peak 4 and peak 5 of the HPLC separation, while there is a really strong effect, cytotoxic effect, in both murine cell line, in all the various fractions. And if we look at the human tumor cell line, we see that the cytotoxicity is lower with respect to the murine cell lines, and it seems more like a cytostatic effect. In fact, we can see that also at very low concentrations, like for example here, peak 5 at 1 microgram per ml, in both cell lines, we still have a lower number of cells with respect to the control. So it seems like there is an arrest of the cell growth. So the next question was to try to understand which kind of cell death the cells were undergoing during this process of cytotoxicity. And we performed 2 types of experiments, an LADH assessment to measure the liver, the degree of necrosis, and afterwards an annexin dye measurement to evaluate the percentage of apoptosis. And we performed the test again on the 4 tumor cell lines, to murine and to human, and we can see that in the final step of purification, again, we have peak 4 of the HPLC separation. There is a very low degree of necrosis, and very low degrees of LDH release from the cells in all cell lines. There is just a little bit more release in this cell line here. MDA cells are human breast carcinoma, but the other cell line, we can confidently say that the necrosis phenomenon is really at low degree. So when we look at the apoptosis, we observe that there is a high degree of apoptosis in this cell that are treated with a purified protein, with the chondrocene, and it's more clear in the murine cell line, in the murine tumorigenic cell line, this one and this one here, when we can see that there is a late apoptotic phenomenon here and here. While in the human cell line, here and here, we have a certain degree of apoptosis, and is seen more an early apoptotic phenomenon at 6 hours of incubation. So of course, it's much more evident the cytotoxic effect on the murine cell line with respect to the human cell line, as I already said. Okay, so the last question that we wanted to answer was if it was possible to somehow understand the mechanism of action of this chondrocene protein, this new protein that we identified in this Chondrosia reniformis extract. So since, as I told you before, there is a domain in the chondrocene that is very similar to the ryanodine receptor calcium channel, and this ryanodine receptor calcium channel is a channel that is able to be opened upon particular stimuli in the cell, and is able to release calcium in the cytoplasm of the cell. We wanted to test also if chondrocene, our new protein, was able to induce an intracellular calcium increase that could could be one of the reasons for the induction of apoptosis. So we performed an experiment measuring the intracellular calcium increase after administration of the P4 purified peak of the HPLC separation. And as you can see here, there is indeed an increase of the intracellular calcium concentration in both the murine two cell lines. In more or less 1 hour acquisition. Here you can see the graph that is the slope per minute ratio of this curve here, and the colored histograms here clearly demonstrate that there is a great increase of the calcium inside the cell. So the next question is, where does this calcium come from? From intracellular stores or from the extracellular medium by opening of some channels? So we removed the extracellular calcium from the medium, and we again performed the experiment. And this is the result are the white histogram here and here in this other cell line. As you can see, removing the calcium from the extracellular medium, we have a complete abolishment of the intracellular calcium rise. So chondrocene is somehow able to open some channel, some receptor on the plasma membrane of the cells, and to allow the calcium entry. Another important mediator of apoptosis is the rise of reactive oxygen species, and these reactive oxygen species can rise after a stressful stimulus inside the cells, but they also can rise after intracellular calcium rise. So we tested again this other possibility of chondrocene, determining an increase of reactive oxygen species in the two cell line, and we could observe, these are the colored histograms again, that in the presence of chondrocene, we observe an intracellular reactive oxygen species rise in both cell lines analyzed here. And again, we performed the experiment in the absence of calcium, of extracellular calcium, and we could see that again this intracellular rise of reactive oxygen species was significantly impaired. And this means that the ROS production due to the administration of chondrocene is consequent to the calcium rise in the cytoplasm of the cell. Okay. So finally, to give a summary of this part of the SMARTED project research, we can say that we have identified and described a new type of cytotoxic protein from the marine sponge Chondrocene reniformis. That this protein really shows unique features. It shows an high homology with a region of the ryanodine receptor calcium channel, and an homology to the domain of the beta sheet of the defensine. The domains of the defensine are very well known in the field of the toxin of protein origin, while the presence of a domain of the ryanodine receptor channel is a new entry in this family of protein causing toxicity in the cells. The action of the chondrocene, this new protein that we identified, is mediated by extracellular calcium intake, which is followed by the cytoplasmic ROS overproduction. We hypothesized the involvement of a particular voltage-dependent calcium channel on the plasma membrane of the cells, which is called CAV 1.3, because this channel has been demonstrated to physically interact with the ryanodine receptor calcium channel and to be opened by this physical interaction. So since there is this strong similarity between chondrocene, between one domain of chondrocene and the domain of the ryanodine receptor channel, we hypothesized that also chondrocene could act in this way, allowing the activation of this voltage-dependent channel and allowing the calcium entry in the cells and causing afterwards the apoptosis. So it would be very interesting to develop this project more, for example, by studying the cytotoxic effect of chondrocene in other type of cells. It has been reported in the literature that, for example, this CAD 1.3 voltage-dependent channel is expressed in neurons. So one possibility could be to study the effect of chondrocene on tumor cell lines of neuronal origin, and this would be really an interesting development of this project. Okay, I'm finished. I leave you to Marco for the conclusion remarks of our project, and thank you for your attention.
Okay, thank you, Sonia, and Marco.
Okay.
I remove my screen. Okay.
Now I have. I try to. Okay, I share the screen. No. Okay, thank you. So, do you see the screen?
Yes, Marco.
Okay, so thank you. Only some final remarks and some very short discussion on the results of this project. Basically, we have obtained almost all the predicted results. And we are happy because we have obtained very nice results from different points of view. Firstly, advancement of knowledge. We have seen that now we have described the very peculiar forms of marine collagen, because the forms of marine molecular feature of the marine collagen chondrosia is very particular. Then we have found a new family of marine toxin, because chondrocene is the first example of a new family of marine toxin, just due to this particular homology with the ryanodine receptor. And this is published, while the other results are we are preparing the papers. But these results are not only scientific results, but has application. We have seen that with this joint project, we have now new approaches for sponge biomass production, and we can obtain new material collagen-based, because this peculiar collagen give peculiar materials interesting for both cosmetics and for regenerative medicine. And this new kind of toxin opened the door to deeper analysis for anti-tumor properties or for new molecular tools of study in neuroscience. These results have been published in Marine Drugs, and we have obtained also the cover of Marine Drugs. And we have also organized a special issue of marine-derived products for biomedicine. And again, we have the cover also of the special issue. So chondrosia in this moment is in two cover of Marine Drug. This is important journal in the field of marine biotechnology. Then technology transfer. The results of this project has attracted the attention of two companies. One interested in the follow-up of the results in mariculture. This is a company of innovative aquaculture technology. And the other company is interested in the production of biomedical devices based on collagen. These two companies are important companies in the field. After this, another topic of relevance for this project, we have strengthened the relationships between Italian and Israeli scientists. We are doing joint research. We are strengthening friendship. There are strengthened friendship between University of Tel Aviv and University of Genova. Professor Mikhailan now is involved in the scientific board of marine science PhD course of the Centro del Mare of our university. This is the largest, the biggest PhD course of marine science in Italy, more than 20 fellowship each year. Undergraduate training, one master thesis of this topic comes from this project. And then dissemination and society sensibilization. We have involved the API and we thanks this group of friends in the dissemination activities. And as we have learned last year, we have performed one conference in one of the most relevant events of scientific dissemination here in Liguria, that is the Festival del Mare. So I conclude just suggesting new frontiers. We have started studying sponge, and sponge inspired the figure of SpongeBob. Everybody knows SpongeBob. But Chondrocene behave like Barba Papa. I think everybody knows, not only in Italy, because it's an international cartoon. And now we have an open question. We are it is very interesting. It will be very interesting to understand which will be the molecular basis of Chondrocene barbatrucco. Barbatrucco is a neologism normally cited in Italian language, that is Barba Papa magic trick. And Chondrocene is the only sponge that is able to do some barbatrucco. Now, our next step is to better explain this kind of barbatrucco. And thanks to this project also, we will have the chance to. So finally, I thanks also the group. This is the Italian group that cooperate in this project, also a group of chemists, Maila Castellano, and our friends Gianluca, Annalisa, and Enrico, Gianluca Damonte, Annalisa Salis, Enrico Millo from the group of mass spectrometrists, and Marina, Caterina, Eleonora, our PhD students, and Sonia. I thank you for the time, and in this slide we have our building and of course the contact for any question. So thank you, and I have...
Thank you very much, Marco, for your conclusion, and all the speakers for the very interesting presentation of this project. And now I think that we have time for questions, also for the people in connection on YouTube channel, if there is some question.
Yes, I would have, if I can.
Yes.
Pronto? Pronto? È difficile questa domanda, come fanno a sapere chi ha scritto nel 2638 come imbarazzo? Ma pronto?
Pronto?
Chi è?
Sono Antonio.
What is happening?
I don't know.
I think some hackering of the site, YouTube.
Yes. Okay. So,
okay, Valerio.
Yes. First of all, Marco, this is definitely excellent technology research. I can say it without any hesitation. So really compliments for your great approach. I had a question for Mika. It is something also preparatory for some further projects. That is, well, Micha, sorry. After your presentation, I really feel I love spongy. Besides various applications you mentioned, including hair of health of hairs, that I care a lot, of course, there is some anti-inflammatory effect that you can forecast from species of sponges, because we are going to submit a project on anti-inflammatory compounds, and I was wondering if in the future we could cooperate on such topics. That would be great, because I think we work on similar topics. You will see also tomorrow during our presentations with apoptosis and other stuff, although we are working on shrimps. But I know that Maria Costantini, with which we cooperate a lot, she's a spongy, definitely a spongy person. She has spongy in the soul. So she would be more than happy, I'm sure, to cooperate with your group. So the question is if you know about anti-inflammatory effects from these or other species of sponge.
The answer in short is I don't remember exactly, but everything you will call anti-fouling, antibacterial, antiviral, anti-tumor, anti-psychosis, anti-pain, everything you will find. It's amazing. First of all, it's a huge arsenal of more than 9,000 species. Now, each one is a hotel of a microbiota, rich microbiota, fungi, bacteria, archaea. So they really have everything. We just need to find the proper bioassay and then go and hunt for the correct compound or compounds. There is for sure, or they are in plural for sure.
May I add something about this?
Yes, yes.
I just wanted to underline that about collagen from chondrosia, we recently published the paper on the antioxidant and anti-inflammatory properties of peptide derived from collagens of chondrosia. So there is an anti-inflammatory activity, at least from the peptides derived of collagen digestion of chondrosia.
Great. So if so, I think in the next days, but we could start the discussion with Marco tomorrow during the round table at 11:30. Yes. I think with Maria we could have some proposals for you. Yeah. We will decide. Yeah. But that would be great to merge some groups working on marine biotechnologies like our groups. So I think we could enforce our power if we work together. Okay. Thank you so much.
There are other questions, because also I have a question for Mika. There are other questions? No, in the meantime, just I profit of this time. Mika, I have just a question for you about the Sarcodrasus spinolosus, the other sponges that you showed. And just in this last year, we collected some sponges in the Faro Lake in Messina, in Sicily, in collaboration with a colleague that is in the Stazione Zoologica in Messina. And we found in this very particular environment these sponges, the Sarcodragus spinolosus. And now
lake?
Yes, it's a salt lake.
Okay.
It's a very particular environment, and we found these sponges. We characterized with the collaboration of Marco Bertolino, that is a colleague of Marco Giovine, that is in Genoa University, and we identified by molecular and morphological point of view these sponges. And now we have a PhD project also in collaboration with Valerio, in which we are trying to understand if from these sponges there is some interesting compounds from biotechnological applications. So if I understand well from your presentation, you find also collagen, you isolate collagen from Sarcodragus.
Most Sarcodragus, if you look at it, it's collagen. What you see is collagen. And in between some cells.
We have isolated collagen from Sarcodragus already.
Huge amount.
In Sarcodragus we found very peculiar fiber, very fiber made of collagen. We performed some analysis, proteomic analysis, and we check that at the molecular level this fiber are made of collagen, and we try to cast the membrane using this, but the Sarcodragus fetidus, not spinosus. I don't know if the same, but it's another species. And from this species, I have obtained, using the same, more or less the same protocol, by dissolving the fiber, in water are released a lot of this kind of very, very small fiber, but it's possible to see under optic microscopy, not at the electronic microscopy, are larger than the fiber from chondrosia, but are made of collagen and are very interesting, and in this case, also in this case, the result of biocompatibility.
Okay, thank you very much. There are some other questions?
Maria, besides questions, I wanted to add that this morning, in the very interesting session of the other project, according to my son, we had more than 200 registered visitors for our Congress. So, I mean, probably they were students from university, but considering that the topic is very specialistic, I think this can be a good result. And probably tomorrow we can communicate how many persons were listening this afternoon. Okay.
Okay. Other questions, or otherwise we can close these sections. And I think that we will see tomorrow morning at 10 o'clock in the morning. Okay?
Thank you all.
Thank you very much for all. Thank you very much.
Thank you.
For
Bye-bye.
Bye.
Bye.
Bye. Bye. Thank you very much.
Thank you.